seminar will be a really special edition seminar, which is all about trainees. And we are planning on another training series in the coming semester in the spring. And we might have 3 more in the summertime. So the trainee seminar will all be hosted and led by the trainees. So I'm going to point to Evan and you can start. And then also Jack will be introducing the speakers.
Okay. Thank you very much, Yanjun. Can I share on the Zoom for a second?
Oh, of course, yeah.
I'm going to try to share just a quick slide, and then I'll be quiet, I promise. Let's see.
Can people on the Zoom hear what Evan is talking about? I want to make sure that you can step closer to the mic.
Yeah, I can move up.
Or want to keep, yeah.
Screen sharing closed. Started screen sharing.
Can people hear Evan?
Can anyone hear me on Zoom?
Okay, great. We got
confirmed.
confirmed.
That's good. So you know why screen sharing is paused. Okay. Technical difficulties. Hello, everybody. My name is Evan. I have a slide. You cannot see it. Don't worry about it. This is the first of the trainee seminar series that DBMI is putting on. The format for this is a little different than the standard ones. Each speaker is going to have 20 minutes, and they can use that time however they'd like. These don't have to be data complete stories. You don't need to see the conclusion of every single experiment. This is to get up and to practice talking in front of an audience, which, it's stressful. I feel a little bit of butterflies right now. So getting people up more often in front of audiences so they can share ideas. For trainees in the room, if this is something that sounds really cool to you, talk to me. I'll try and share a thing at the end of this, but we might be able to set up some sort of informal group where we can get together and do this more often. But this is the official one, so I will get off the stage and let the actual presenters present. Okay, Jack.
Hi, everyone. So thanks to Evan for getting this going and organizing the student trainee seminar series. I also want to thank the department for their support on this and all of our administrative staff who's been very flexible in accommodating this sort of format. And I want to thank the students who volunteered to speak. So first up, we have Grace Akatsu. They are a MD PhD student in Casey Green's lab, and they are going to be talking about characterizing cell types in ovarian carcinomas using transcriptomics. And so Grace, want to start us off?
Thank you so much for having me. Thank you. I am relieved to hear that there is no expectation for a lot of data. This will be mostly prospective. I'd like to share the plan I have. I just started my PhD a couple of months ago. The plan I have for what research I want to do, would love to hear questions, ideas, suggestions. Okay, so, and also I'm gonna just keep an eye on the time. Okay. Oh my. Okay, so as mentioned, I'm an MD PhD student. So if you will indulge me, I would like to start with a clinical vignette. This is based on a patient I saw last year at Denver Health. Anonymized, but based on. Okay, so a 59-year-old woman with a past medical history of symptomatic menopause treated with hormone replacement therapy, as well as class 2 obesity, presents to her PCP with 2 months of abdominal pain, bloating, urinary urgency, and fatigue. In the clinic, her vital signs are within normal limits, and on exam, she has right lower quadrant abdominal tenderness, and there's a palpable right adnexal mass. Her physician orders some studies and as well as a transvaginal ultrasound. So show of hands, has anyone seen a transvaginal ultrasound, what that looks like? Yeah, a couple people. So here, for the people who haven't, that's totally fine. Here's what a normal right ovary looks like on transvaginal ultrasound. This is the ovary, and these anechoic circles are follicles. And this is something like what the patient's right ovary looked like. So as you can see, much bigger, there's this large heterogeneous mass. And I won't get into too much detail, but there's this transvaginal ultrasound classification system called ORADS, basically how, if you see an ovarian mass, how likely is it that it's malignant? Her mass classifies as ORADS 5, greater than 50% chance of malignancy. And per guidelines, she's referred to gyn-onc and undergoes debulking surgery. And surgical pathology reveals high-grade serous ovarian carcinoma. And that is the disease that I will be talking about today. And I'll be using transcriptomic data from patient samples. And so in order to kind of stay connected, I would like to show an image of a surgical specimen. If that's not what you're trying to look at during your lunch, I totally understand. Feel free to avert your eyes and I will let you know when it is safe to look back. But here's an image of what the surgical specimen might look like. So just to orient you, this is the uterus, this is the cervix, this is the left fallopian tube, fimbriae, and wait, can you see my, okay, yeah, and the ovary. And then over here, this is the right fallopian tube, and this is the mass that we saw on transvaginal ultrasound. This all up here is omentum, and these white dots are likely metastatic disease. So, okay, you're welcome to look back if you had looked away. So what is high-grade serous ovarian carcinoma? We can break down the words in the long title. So high-grade, that's just a histopathological classification. Basically, when you look at the tumor under a microscope, how ugly does it look? How different from normal tissue does it look? Serous, this is kind of more of a historical histological subtype. There's 4 subtypes of epithelial ovarian cancer, and serous is one of the 4, the most common. Ovarian, possibly a misnomer. We originally thought that the source of the cancerous cells was the ovary, and now we know it's probably more likely from the fallopian epithelium, but the name remains. And carcinoma, that just means a cancer that originates from epithelial cells in opposition to sarcoma, which is from connective tissue. So, okay, high-grade serous ovarian carcinoma. This is the most common form of ovarian cancer. 90% of ovarian cancer deaths are due to this cancer. And unfortunately, the 5-year survival rate is 40%. And this 5-year survival rate remains essentially unchanged since the 80s. So thank you for bearing with me with that introduction. Now to kind of go into the rationale for why I am proposing this research. So tumor cell type composition is known to correlate with high-grade serous ovarian carcinoma clinical outcomes. So this is a study that was looking at aggregating a bunch of data. It looked at the proportions of immune cells and the correlation with overall survival and progression-free survival. And so as you can see, it looks like macrophages are maybe slightly protective if you have more of them, whereas the CD4 memory T cells maybe are a more ominous sign. So, and this and other studies have shown that the tumor cell type composition is really important to understand because it's clinically relevant. However, our current understanding of high-grade serous ovarian carcinoma cell composition is based solely on single-cell RNA-seq, which is a technology that I'm sure everyone is aware of. It won Nature Method of the Year in 2015-ish. Like, it's really a paradigm-shifting technology. And it has its own technical limitations. And because we have relied solely on this technology, our understanding is colored through the lens of those technical limitations. So this is a figure from a paper that was looking at human adipose tissue. So they took one sample and did single nucleus RNA-seq and whole cell, also known as single cell RNA-seq, on the same sample. And as you can see, there are certain cell types that are captured with single nucleus RNA-seq, but are absent in the single cell RNA-seq data. And this has been shown in other tissue types as well. The reason for this is that for single cell RNA-seq, has anyone prepared a single cell RNA-seq library before? Yeah, some people. I did a long time ago, not very well. But you have to dissociate the cells in order to put them in the droplets to feed it through the machine. And that is a stressful process, and delicate cells cannot survive this process. So I hypothesize that technical limitations unique to single cell RNA-seq pose a significant barrier to our understanding of the high-grade serous ovarian carcinoma cellular milieu. And I guess I should also mention to my knowledge, there's one publicly available single nucleus RNA-seq data set for this cancer type available, and nothing more. So here is maybe a slightly overwhelming table, but we'll go row by row. So the top row, this is what I've mentioned, single cell RNA-seq, lots of benefits. However, it is only able to capture some cell types. The next row down, single nucleus RNA-seq, it is able to capture all the cell types because it doesn't involve dissociation. However, by definition, you're not capturing the cytoplasmic RNA, so you're not getting a complete look at the transcriptomic picture. And then spatial transcriptomics, have people worked with, heard of spatial transcriptomic data? Maybe a little bit. There's 2 different kinds. So probe-based spatial transcriptomics, this is like MERFISH or Xenium. This is where you're using, it's kind of like a microarray. You have probes for specific transcript sequences that you're looking at, and then you do like imaging to like look at where the transcripts are, and then you can approximate, kind of a subcellular-ish resolution and assign transcripts to cells via imaging. Because it is not next generation sequencing, you're not capturing all of the genes, you're just capturing the transcripts you have a probe for. And the resolution is not as high as single cell or single nucleus because you might be misassigning transcripts to cells because you're just looking at one plane. Sequencing-based spatial RNA-seq, that is like Visium. That's where you have these like pixels or spots that you are performing next-gen sequencing on. So you're capturing all the genes, but each spot has multiple cells or can have multiple cells. And then finally, bulk RNA sequencing, as we know, the resolution is very low. You're just looking at all the transcripts in the whole sample. Okay, that was kind of a lot. Any questions so far on the setup, the rationale? Okay, so to just summarize my rationale, tumor cell type composition in high-grade serous ovarian carcinoma is correlated with clinical outcomes. A detailed, accurate, and complete understanding of the tumor microenvironment is very valuable information in terms of advancing treatment. However, technical limitations unique to RNA-seq modalities, specifically single-cell RNA-seq, have posed a significant barrier to this goal. So what I am planning to do, there are two aims that I'm planning. The first one is to just compare the benefits and technical limitations of different transcriptomic modalities in determining the cell type composition, which so far has only been done with single-cell RNA-seq. So just to orient in this diagram, so this is a human high-grade serous ovarian carcinoma sample, and this is the true cell type composition. So you can see it. This is the pie chart of the true cell type composition. And if we use single-cell RNA-seq, in this example, it's not capturing the purple cells. So you get this kind of calculated cell type composition. Single nucleus RNA-seq, you're missing the cytoplasmic RNA, so maybe something like this. Probe-based spatial RNA-seq, and sequencing-based RNA-seq. So I hypothesize that the transcriptomic modality you choose will affect the calculated cell type proportion, and that possibly looking at these different modalities will uncover cell types not currently existing in current atlases of high-grade serous ovarian carcinoma. And as I mentioned, there is not there's one single nucleus RNA-seq data currently available, and so our collaborators at the University of Utah are creating some paired single cell and single nucleus RNA-seq, so taking one high-grade serous ovarian carcinoma patient sample and performing both single nucleus and single cell RNA-seq on that one sample. Okay, and then aim 2, I would like to generate a model to identify the impacts of specific technical factors to create a novel deconvolution method to apply to bulk RNA-seq data. So when I see deconvolution, that is the process of computationally estimating cell type proportions from bulk RNA-seq data. And currently the deconvolution methods are trained on single cell RNA-seq pseudobulks. So it presumes that single cell RNA-seq pseudobulks accurately approximate bulk RNA-seq data, which I would argue is not necessarily capturing the whole picture. So I've already talked about aim 1. For aim 2, I would like to adapt Buddy, which is a variational autoencoder created by Natalie, a former postdoc in the lab, and which has these modular encoders and latent spaces to capture different sources of variation. And I would like to train it to capture sources of variation, the specific technical factors that I've talked about, for instance, presence or absence of cytoplasmic RNA, probe-based sequencing versus next-gen sequencing, et cetera. Yeah, okay. And so preliminary results, I have one slide of extremely preliminary results, but I'm just starting to get my arms around this paired single cell and single nucleus RNA-seq data that I have mentioned. And so this is from the same patient sample. And as you can see, it looks like the single nucleus RNA-seq is capturing some clusters that may represent cell types that are not captured by single cell RNA-seq, analysis still pending. Okay, so in conclusion, I've shown, or the literature has shown that high-grade serous ovarian carcinoma cell type composition is really important to understand. correlates with clinical outcomes, and detailed, accurate, and complete understanding of the cellular milieu has a lot of potential applications in terms of personalized treatment, understanding disease prognosis for individuals, identifying novel treatment strategies. However, technical limitations unique to RNA-seq modalities have posed a significant barrier to this goal. So overall, I would like to synthesize data from multiple transcriptomic modalities and use each to kind of counterbalance each other's technical limitations and arrive at a better understanding. Thank you very much, and I'm happy to take questions. Yeah.
Thank you, Grace. This is not a question about the science, but rather I just wanted to share an observation that I really like the way you set up the stage with your clinical vignettes and the way that you, at least to me, it was inspiring just to see that as a background to why you're posing those questions.
Thank
you. I just wanted to say good job.
Thank you. I appreciate it. Yeah. Yeah.
Obviously, awesome talk. Very important, very interesting work. I totally get why it is important to focus on one specific cancer so that you have complete coverage across all these data types and so forth. But can you talk a little bit about how you're thinking, how to take the lessons that you've learned from this particular cancer and then maybe apply it to other cancers or even other data sets from other conditions? How do you know that things that you learn will generalize?
That is a really good question. And I think the dream would be to create a pipeline where you can put in different data types from the same tissue with cell type annotations. And then ideally the pipeline would create the pseudobulks, train the model, create a deconvolution method. I think the hiccup to that is like having a well cell type annotated data set from multiple different modalities. Yeah, but I do think the nice thing is that using pseudobulks from different transcriptomic modalities, you have a ground truth to be able to train on, potentially. Yeah.
Is there like a ground truth for like the proportion of cell types from these patients and things?
Yeah, that is a good question. For my work, I will be assuming yes. But I think cell type annotation is its own kind of like can of worms. Yeah. Yeah.
Very nice talk with you. Sorry if I missed this, but what would the final output of whatever you're synthesizing? What would that be? Like, would it be like a condensed representation that's formed from all different modalities to get some feature vector or something for a particular sample? Or like, how are those all being integrated together?
Oh, that's a really good question. And maybe I was unclear. The idea is to eventually be able to make a deconvolution method so you have a vector of proportions for different cell types from a bulk RNA-seq data, and possibly that could be used as a biomarker or at least to characterize better. Yeah. Thank you. Yeah.
In your single cell and sort of like single nuclei focused analyses, I might have missed this. Are you planning to interrogate patient-based data or cell line data? And if it's patient-based, how many sort of samples do you have to actually annotate these cell types?
But to make sure I'm understanding your question correctly, the question is, wait, I don't, what do you mean by cell type data or cell line data?
So where does the data come from? Does it come from a person or does it come from like different like 3D models or co-cultures or whatnot?
I understand.
Yeah.
That is a really good question. No, I appreciate it. It's coming from people. I think there are 12 patients.
Okay.
Yeah.
Do you have matched clinical information for them?
That's a good question. I think so. Okay. Thank you.
Okay, next up, we have Kat Terwelp, who is a second-year comp bio student in the Robbie Lab, who is going to be talking to us today about horizontal gene transfer and microbial social networks. Okay. Go Kat.
Yes, I changed the title. It's now listening in on microbial social networks. And the whole thing is in parentheses. So we will get on with the presentation once I remember how to use things. Okay, cool. Why am I presenting today? Jack asked me. Well, also, this is going to be a proposal talk, not a data talk like Grace. I managed to get 2 slides of data from yesterday's work, so hot off the press for you guys. But I will have a couple questions for you at the end, mainly because I want to start working towards like the NSF GRFP. Hopefully the solicitation will come out, but the deadlines are available. And also for comps later next year. So what is horizontal gene transfer? Well, if you're more human oriented, you probably know what vertical gene transfer is. That's inheritance from parent to child, genetic information moving that way. Horizontal is, in comparison to this, going from donor giving its genetic information to a recipient. And so these donor and recipient bacterial cells don't necessarily have to be genetically related, right? So that's a very cool thing that bacteria can do. Why does this matter? Well, if you eukaryotic focus, it is how we got mitochondria. If you're microbial focus, it's a big important factor of microbial evolution. It drives niche adaptations, such as perhaps gaining the ability here's a little cartoon, to be resistant to certain antibiotics. These are often genes that are transferred horizontally. So that is an important human health related phenotype of bacteria. So how does this happen? There's 3 canonical methods, and there's a bunch of other methods, but we'll focus on these 3 for now. Transduction, viruses that infect bacteria can sometimes make a little mistake where they package the first bacteria they infected's genome instead of their viral genome, and then they transfer that into their next host. So that's transduction. Transformation is bacteria's ability to take DNA from the environment. And conjugation. This is where 2 bacteria form a pilus, which is like a tube that enables the transfer of DNA through that tube from the donor to recipient. So those are the 3 methods that people generally are taught, but there are others, but I will not go through them. And then how do we detect horizontal gene transfer? I'm going to focus on computational methods because I'm in the computational program and I'm not going to be touching a pipette for 4 years. But in general, wet lab methods are focused on the specific type of horizontal gene transfer, and I'm more interested in all the types, right? So here are 3 main classes of methods that we can use to detect horizontal gene transfer. The first that I'll talk about is compositional. So this is basically, you take a genome and you look at it and you're like, this section of DNA does not look similar to the rest of the genome. Alien DNA, it is probably horizontally gene transferred. Phylogenetic is when you take a species tree. This kind of represents vertical transfer of information, and you compare it to individual gene trees. So like for each gene in a bacteria, you construct a phylogenetic tree. And if there are changes between that gene tree and the species tree, you can hypothesize, okay, this is an example of horizontal gene transfer. And then taxonomic is kind of like trying to skip that phylogenetic tree building step. So instead, it takes a gene and blasts it against a database of bacterial genes. And it's like, okay, the top hits are from taxonomically distant bacteria. So bacteria that are unrelated to this organism. So that's probably horizontal gene transfer. So those are the 3 main types that we use. And then since I am interested in scaling this up, which ones are scalable? Compositional and taxonomic are definitely phylogenetic iffy. But there are reasons to use that phylogenetic method. First, recency. So some horizontal gene transfer happens very recently in the evolutionary history of these bacteria, and those are, they tend to be very similar to the donor organism. That DNA tends to be more similar to the donor organism than the recipient. And as time continues, the piece of DNA that's been incorporated into this recipient genome starts to accumulate mutations that make it look more like the recipient, right? So compositional, when you're looking for alien DNA, it works best with recent transfers. Phylogenetic and taxonomic can work with both. Directionality. So this is just my way of saying, can we tell who's the donor and who's the recipient? Compositional, no. You just know alien. You don't know where. Phylogenetic, yes, you can tell exactly who the donor is and what gene is being given to the recipient. So you know the donor and the recipient and the direction of that. Taxonomic, you can have a kind of guess of the partners in the horizontal gene transfer, but you do not know the direction, who's the donor and who's the recipient. So large scale patterns of horizontal gene transfer. This is a pretty picture from a paper that did about 6,000 genomes of bacterial genomes and found, I think, over 140,000 transfers using a more compositional heuristic approach. And the colors of these dots, each representing a genome, is based on the taxonomic group of that bacteria. So you might be seeing some patterns, but let me lay them out for you. Phylogenetics is a huge contributor of horizontal gene transfer. As organisms are more phylogenetically distant, you have less transfers. They're just not as similar, and maybe they don't like each other. Ecology. This is basically bacteria that co-occur, are close to each other, are more likely to have higher rates of transfers. This kind of makes sense since I went through those methods. Those tend to be kind of requiring contact or requiring close proximity, right? So it makes sense that it's ecologically, ecology helps drive horizontal gene transfer. Also, as a side note, hosts like humans, host-associated bacteria have much higher rates of transfer. This also makes sense when you think about the environment of certain places in the host, like the gut, which are super dense microbial communities, basically like cities, when you compare it to like environmental microbes that are floating many, many cell lengths away from each other. Lastly, gene functions. These can help predict horizontal gene transfers, and also certain gene functions are more common. For example, antibiotic resistance is more common in recent transfers. And defense, so defending against alien DNA because it can be poisonous, is also a gene function that helps predict horizontal gene transfer and is also more common in transfers. So all of this previous literature is looking at large-scale patterns of horizontal gene transfer. And that's like using it, like looking at predictors. And I was thinking, what if we use horizontal gene transfer as the predictor instead for bacterial phenotypes? So why am I interested in bacterial phenotypes? It's because I'm in the JROVI lab. So what exactly does that look like normally? Normally, we like use machine learning algorithms where we take, like, for example, for species A, we take genomes and we find some genomic information like the genes they have, and we take known phenotypes of each of those genomes, and we use machine learning to find the rules or what genes help predict that phenotype. But the issue is when you try to put those rules and apply them to species B. As you can imagine, this doesn't always work out, especially as species B becomes more like distantly related to species A, because some of those genes may not even appear in species B's genome. So I was thinking, horizontal gene transfer seems like a great potential biomarker of shared ecology and potentially shared function. So what if we could use horizontal gene transfer networks to predict bacterial phenotypes across diverse species? And so here's my simplified diagram of what I think that could look like. This is if everything works wonderfully. And this is just like a simple example where we have species A, species B, and they're connected by horizontal gene transfer. And you can see, okay, this A is connected to B. I shouldn't point since there's Zoom people. Oh, well, this has disappeared, but that's okay. A in the middle, right here, is connected to B, and they have a shared phenotype. So that would be enabling predictions across species barriers. So I do have results, as I alluded to. But first, methods of how I did that. I decided to do phylogenetics because it's kind of the gold standard and it gives me the most like detailed information, which I like. So I take genomes and I construct pangenomes. What are pangenomes? It's just taking all those genomes, finding all the genes, and you find out which ones are core, genes that are shared across all these genomes, and which ones are variable or accessory genes, which are not shared. Some bacteria have them, some don't. And in this case, I try to filter out these genes to ones that at least 5 of the isolate genomes have. And then I build my phylogenetic trees. For the core gene, I use that. I concatenate them all together to make the species tree. This represents my vertical transmission of genetic material. And then I create my individual gene trees with both the core and the variable genes. Lastly, I use Ranger DTL, which reconciles the differences between the two trees and identifies horizontal gene transfer events. So that's the general pipeline. Now for some cool results using core diagrams because they're pretty. So in this light blue is highlight cyanobacteria. So cyanobacteria are kind of like algae. They are photosynthetic organisms that live in the ocean. So the highlight ones live closer to the surface and the low light ones live a little deeper down. Those are the two main ecotypes for Prochlorococcus or the cyanobacteria I'm looking at. And as you can see, the highlight genomes transfer with the highlight genomes and the low light genomes transfer with the low light genomes. So they generally do not like to transfer across those barriers. So they're a little bit not liking to share. Now this one is a little bit more complicated. I got a little bit more colors going on and a little bit more exchange of material outside of their color-coded thing. So we have Staphylococcus aureus genomes, which is a like MRSA, so pathogen, not always great to have. And I took 175 of those genomes with known hosts that they were taken from. So we have human, camel, cow, pig, and monkey, as I've drawn. And you can see like human tends to transfer with other human isolates, monkey with other monkey isolates, pig mostly with pig isolates, except also with human, it appears. So it's a little bit more messy when I look at a more complicated phenotype, which is what I expect, and also motivates actually using machine learning. Like I don't need to use machine learning for this. I can easily tell. This is a little harder. So I think this is a good example of how horizontal gene transfer networks do include important information that we could potentially use for these models. But although this is promising, maybe, there are some obstacles I want to go through today. Horizontal gene transfer detection is not solved. That's why there's like 3 main methods, and in each of those methods are many different papers for the new best tool to use, right? So how robust are these how robust would my machine learning results be if I use different methods? So I plan to compare results between different detection methods and also use multiple methods to make my networks. Okay, potential obstacle number 2, how will scientists interpret and use these results? Like, if I have a nice picture, that is helpful, but how do they actually use this? I plan on using, at first, supervised interpretable machine learning models based on like adjacency matrices. So that would let me maybe identify important neighbors that may lead to a certain phenotype, like, okay, these bacteria are pairs, and if I know this neighbor is a pair, okay, they probably transferred antibiotic resistance, things like that. And then for like, if I want to get into graph, like neural network machine learning, I could try to use something like GraphLine to help interpret what the most important features are. So that's how I plan to handle interpretability challenges. And then lastly, are these networks more informative than simply using a phylogenetic tree, right? A huge predictor of horizontal gene transfer is phylogenetics, right? So I think it's important to compare both if I want to claim that horizontal gene transfer networks are worth the extra like 10 plus hours of processing after you make a phylogenetic tree. So I plan on comparing these, and that's kind of what my 2 like questions or like my aims I plan on doing for this kind of project is comparing horizontal gene transfer machine learning models to ones built with just capturing the vertical genetic transfer of information. So broader impacts, I think it would enable us to identify bacteria that might be bridges that enable species to gain the ability of antibiotic resistance or other human-related things like host switching. Now this pathogen can infect humans, that's bad, things like that. So, or niche adaptation, and might help with bioengineering microbial communities for positive things like bioremediation, finding out ways to introduce genes into those communities to maybe help like degrade plastics, things like that. So questions and feedback. Hopefully that wasn't too fast. I know I talked really fast.
So in your machine learning algorithm, are you going to consider how physically close in the environment these bacteria might be? So the likelihood of interaction between the two, like the cyanobacteria are probably not going to interact because they're in like two different spaces. Right?
Yeah.
But if you like sample in between, maybe you see more transfer.
Yeah, I think if the metadata exists for that, that would be wonderful. I might be able to approximate that kind of stuff using like, for example, the paper that I used for like saying co-occurrences of factor of horizontal gene transfer, they use publicly available microbiome data. So I could see what like, and a lot of that's going to be 16S, which only gives you maybe species or genus, but you can kind of get, okay, this genus is appearing often in the environment and also in humans, things like that. So I could maybe add like the habitat these like genus or species are located in as like part of my machine learning models. Yeah.
I have two questions. One is, what is the unit of the genetic material that's being transferred? Is it like a whole gene, or there are portions of the genetic material that could be transferred? And my second question is, are there any signatures within the transferred material which can say that it is likely to be more transferred or not?
Yes. Okay. So for one, it's like, let me remember. So, okay, the unit of what's actually being transferred. So for my phylogenetic method, it's looking at genes. So it's just going to say if the whole gene is transferred or not. But it's not necessarily a case that it's only one gene that's being transferred. Sometimes multiple genes are transferred all at once. And sometimes, like if it's poorly packaged genome from a virus that messed up, like it could not just be one gene, it could be like part of like a random piece of that bacterial DNA. So that method might not capture that. So that's where using multiple methods might help. And then for the second question, oh gosh, I forgot.
So if there are any signatures within them.
Okay, signatures within the gene that's being transferred. So we know that like recent transfers are enriched for antibiotic resistance and defense genes, so preventing transfers. And maybe older transfers are enriched in metabolic genes, just because these transfers have lasted so long in the genome of these bacteria because they've made some sort of useful thing for that bacteria in that ecological niche. So like metabolic genes make sense in that case. So there are definitely signatures that people have used to try to predict, but how well these signatures align across multiple studies is kind of iffy. Like a lot of them have the antibiotic resistance in the defense, but some of these other like outside of those functions, they're not always like in agreement for these studies.
Can you speak a little bit more like the tree generation for when you're doing the active genes, like the genes, not the species level tree generation? Because for the species level, you're taking something like a 16S gene, right? And you're looking at sequence similarity to get the tree. Mm-hmm. But when you're taking like a core genome and an accessory genome, like how do you generate a tree from those? Is it at like the specific gene level, or do you assemble them into genomes, or how is that?
Yes, it'll be just at the gene level. So like I'll have, Panta will give me like for each gene, the alignments of like across all of the genomes for that gene. So I can use that to construct the trees. So each of the trees is just one gene. Yeah, and then for the species tree, I actually do something that's maybe a little bit more robust than 16S, which is concatenating all of the core genes together. So then I have the like different like alignments for all of those genes. And so that's a little bit better, but anyone who's really into phylogenetics can yell at me. That's fair. I've just gotten into it.
Have you heard of that, or do you use PAML? It's a software for looking at phylogenetic analysis.
I have not.
We have. I don't do it. There's someone in our lab who uses it.
Yeah.
So I could connect you guys if you were interested. But
that would be great.
Use it for looking at evolutionary pharmacogenes across different groups.
Yeah, that would be awesome.
Yeah. I think it seems relevant, but
yeah, I'm in the needing a bunch of tools to try out phase. So with my couple of months in this lab. Yeah, cool. Thank you. I see my over time, but yeah. Also, if you have problems or concerns, you can QR code, send me an email.
Okay, next up we have Ashley Weir, who is a visiting PhD candidate in biology at the University of New South Wales in Sydney, and now is in the final year of their computational PhD at the Walter and Eliza Hall Institute, where they're studying under Dr. Nadia Davidson and Dr. Susan Ramos. Take away, Ashley. Thanks for visiting.
I will.
Speaking.
Just one moment. My slides will not email because, of course, not.
That's okay. I will, or let me send you the Zoom link. Yeah.
So yeah, I'm visiting from the Walter and Eliza Hall Institute in Melbourne, Australia, and I have 3 months left of my PhD. So this is not going to be a proposal. Though that does not mean that it is flawless, as everyone with a PhD will know. So yeah.
Yes, I think so.
And for the context, Ashley is visiting my lab and meeting with a couple of people, so she just here today.
Or you can just type it in maybe on Zoom.
Oh, this is. Sorry about that, Ashley. No, it's all good. This is a part of presenting. Navigating Zoom is important. We all should know how to do it at this point, and yet, okay. So thanks for having me today. I am here to speak about the first aim of my PhD project, characterizing genomic damage in high-grade serous ovarian carcinoma through transcriptomics. And Grace gave us a lovely introduction to high-grade serous, so I don't have to do it, which is just a lovely, coincidence for today. And so the underlying biological problem for this aim, or the motivation for actually trying to characterize genomic damage in high-grade serous through transcriptomics, stems from the fact that 50% of these tumors will have a therapeutically targetable defect in DNA repair, or more specifically, in the homologous recombination repair mechanism for double-stranded DNA damage. So high-grade serous tumors can be classified as being homologous recombination, or HR proficient, whereby double-stranded DNA breaks can be repaired in this relatively error-free fashion. Or they can be classed as homologous recombination deficient, where through various mutations in homologous recombination genes, such as BRCA1 or BRCA2, including epigenetic silencing of these genes as well, a tumor has actually become unable to repair through this relatively error-free mechanism of homologous recombination. And what results is increased genomic scarring. If you have a lot of double-stranded breaks that are repaired in an error-prone manner, you see more scarring. And the reason this is important clinically is because not only do 50% of these cases have a defect in homologous recombination, but this remains one of the only features of high-grade serous that is actually eligible for targeted therapy. So HR deficient tumors, or HRD tumors, are eligible for targeted therapy through a class of compounds called PARP inhibitors. And so it's important to note that the homologous recombination status of a cancer is not actually something that is static, but rather if you have restoration of homologous recombination through reversion mutations or many other mechanisms, you can actually lose your responsiveness to targeted therapy. However, despite the fact that this is not a static process, the way that we detect homologous recombination deficiency in clinical practice largely relies on a HRD score of genomic scarring events derived from whole exome sequencing or SNP arrays. So a HRD score, or a HRD scar score, is an accumulated sum of telomeric allelic imbalances, loss in heterozygosity, or large-scale state transition in copy number. And so once all of these events within a tumor's genome are summed, If the score is more than 42, they are classed as HR deficient, and if the score is less than 42, they are classed as HR proficient. 42 is a debatable number, but I'm not here to question 42. It's worth noting that this score is continuous, and so while it's lovely to view this phenotype as being quite binary, it's kind of not. And yet I will ignore that. And so what I've been able to observe, and the motivation for my project stems from the fact that this remains the gold standard for detecting the HR status of a tumor. There are or were no methods to predict HR status using gene expression, specifically in high-grade serous ovarian cancer, which is important because pan-cancer methods don't tend to generalize well to high-grade serous because it is a very weird cancer. And so I hypothesized that the genomic signal that is used to predict HR status in high-grade serous would translate to the transcriptome, just the central dogma of biology, nothing groundbreaking here. And so the aim of this project was to build a model to predict HR status in high-grade serous ovarian carcinoma using gene expression alone. And so to do so, I accessed data collected by the TCGA across 361 high-grade serous cases. And I randomly split the data into training and testing sets, taking an 80-20 split. I annotated truth labels as I was trying to build a supervised model using the genomic HRD score and that threshold of 42, and then accessed bulk RNA sequencing data for these true HRD and true HRP cases. There are roughly 50,000 genes when we do bulk RNA sequencing. And so I wanted to begin this project by reducing the feature search space. And so I began by doing some feature selection where I filtered out any genes on the Y or mitochondrial chromosomes, non-coding genes, and any genes that were lowly expressed. And what I was left with was roughly 19,000 genes per sample. This is still a very large feature search space, and we expect that many of these genes will be co-expressed and will not necessarily be needed. However, we want to guide our model so that the genes that are selected are truly biologically informative. It also helps like clinician scientists feel a little bit more comfortable when they will maybe hopefully use my tool. So I performed a differential gene expression analysis contrasting the HRD high-grade serous cases to the HRP cases using the standard Limbervoom pipeline. I'm based at the WeHi, and that's where Gordon Smythe is based. He's the person that invented Limmer, and so we all use Limmer. No questions asked. And what I found was that there were just over 2,600 significantly DE genes. When I looked at my top upregulated genes against my top downregulated genes in the HRD group, I was able to appreciate that BRCA1 was one of my most significantly downregulated genes, and it is a known, or mutations in BRCA1 are a known cause of HRD. So this was kind of like a nice positive control, if you will. I then wanted to have some confidence or some understanding of this gene expression signature. And so I performed gene ontology enrichment analysis using Goanna. I don't know, I wasn't in love with these results, to be perfectly honest. Like, they were kind of really frustrating. Like, yeah, DNA damage response comes up, but like organelle, yeah. I didn't like it. I got really frustrated. And one day I decided to ask, well, where are these genes? And I found that the distribution of significantly up and down regulated genes was highly variable based on chromosome arm, even after you normalized for gene density. And so this seemed odd, but also not that odd when we consider the fact that the phenotype I'm looking at involves genomic scarring. And so what I then did was I visualized the location of each gene across each chromosome. And this is showing all genes that I looked at in DE analysis, and they are colored if they are significantly down or upregulated with the false discovery rate set at 0.05. What I was able to appreciate was that there were these regions of purely up or down-regulated expression in our HRD group. However, every now and then there was one gene that kind of went against this current, which is highly selective and very interesting, I think. I then wanted to understand if these regions of transcriptomic change had a potential genomic background, because you would think so. And sure enough, one of my supervisors said, yeah, we did this in the 90s. So that was wonderful. Thankfully, they redid it in 2021 using the TCGA, so there wasn't much point me double checking. But basically, there are 3 regions of known copy number alterations that are dependent on HR status. So HRD cases are known to have an amplification on chromosome 8's Q-arm at band 24, a deletion on chromosome 5's Q-arm, 13, and HRP cases are known to have a 19q12 amplification that's specific to them. Now, none of these copy number changes are truly mutually exclusive. There's some bleed through, likely because HRD and HRP, it's a continuous state, basically. Yeah. And so when I looked at patterns, at my patterns of gene expression, I was able to appreciate that, yes, in this region of chromosome 8, where there is a known HRD amplification, gene expression was significantly upregulated. Yes, at chromosome 5, it was significantly downregulated. And in the HRP group, where we see this amplification at 19, we also see significant downregulation. And so with this, I was quite happy to proceed with these genes as a sort of basis for further feature selection, because I was able to appreciate that they did have some genomic background. And we know that the HRD score has been a really robust way of predicting this phenotype. And so we kind of want to exploit that a little bit without relying on it too heavily. And so I then wanted to scale and transform genes to make sure they were all sort of equally contributing, all had equal potential to contribute to a model. And I used these 2,604 genes as input for a penalized logistic regression model, or to train a model. I tuned L1 and L2 hyperparameters through 5-fold cross-validation, and taking the parameter combination that gave me the maximum area under the curve, what I was left with was an elastic net penalized logistic regression model of 209 genes to predict HR status in high-grade serous, hopefully. I named this tool Identifier. I really like wordplay, so bear with me. To use Identifier, you can have a single high-grade serous sample or a cohort of high-grade serous samples, and all you need is just bulk RNA sequencing. And the output of this model is a discrete HR status in addition to the probability of that sample being HRD, because if we think of it as a continuous phenotype, we kind of want a continuous output as well, though not everyone does, so you still have to give people discrete labels. It's all good to train a model, but then you actually need to prove that it works, which is something that I don't know if I'll ever be fully convinced of personally, but it seems to go okay. So we then looked at our held out test cohort of 73 high-grade serous cases, and we used Identifier to predict their HR status. I was able to appreciate that we had an 85% accuracy, and there was a fairly equal split in terms of the number of cases that were incorrectly predicted against their actual class. But what was really nice to see was when we looked at the probability of a sample within this testing cohort being HRD against their true genomic HRD score, these two variables were quite decently positively correlated. And so the few cases that we did predict incorrectly did tend to sit close to the threshold at 42 or our probability threshold at 0.5, though there were many cases, or there were some cases that Identifier was quite confident were HRD, though their genomic scarring did not yet indicate they were HRD. And so we're following up with these cases to see if we can look at reversions, or if, we can have a little bit more confidence in this. We then wanted to benchmark Identifier against other gene expression signatures. Now, to my knowledge, there were none at this time that had been developed for high-grade serous specifically. And so you still have to benchmark when you're a bioinformatician, regardless. And so I was able to find one gene expression signature that was developed for a breast cancer cohort, another expression-based model, and a pan-cancer model that was recently, it was published the month that I was writing up this, so that was fun. as always. When I looked at the 73 cases in our held out testing cohort, ordering them by their HRD score and also annotating their HR status, I was able to appreciate that identifier predicted the majority of the HRD cases correctly, or identified the majority of cases correctly, as opposed to the Brackenus multiscale HRD and expression HRD signature, which like to call everything HRD. So we had the highest accuracy, precision, and recall, with the exception of expression HRD, but we'll let them have that. We also had the lowest misclassification error. And when I looked at overlap between these expression-based tools in terms of the actual features they use, there was one single gene that overlapped between all four, and it's actually a known HR gene, but it's really poorly studied. So, I mean, I don't have a wet lab, but if anyone wants to follow up on this, I'd really appreciate it. We then wanted to be quite comprehensive in our testing, and so we looked at a completely independent cohort from the Australian Ovarian Cancer Study Group of 87 samples collected from 81 patients. We used Identify to predict their HR status, and we're pleased to see that we had an accuracy of 89% in this cohort. What was really nice, though, is that these samples weren't just taken from primary tumors, but there were also some samples taken from autopsy, and we correctly predicted all of their HR status labels. And I think most importantly, there were some samples taken from normal fallopian tube, and we correctly predicted all of them to be HRP. So our signature is hopefully cancer specific and hopefully isn't too affected by actual sample purity or quality, although I can't 100% guarantee that. We then also did independent testing in a pseudobulk high-grade serous cohort from the Memorial Sloan Kettering Cancer Center of 106 samples collected from 37 patients. We subset this data set down to just the high-grade serous cells. So Grace's work will definitely inform this in future, to look at the different composition of the cells that I have chosen to ignore. And pseudobulking at the patient level, we had noted that 11 of our model's genes were actually missing in this cohort. So we just set their values to 0 and pretended that wasn't a thing. But regardless of this, we were still able to achieve an accuracy of 84%. But as this is single cell data and these samples were taken from various sites throughout the body, we then felt that as if we could actually slightly trust our tool at this point. And we wanted to perform some exploratory analysis. Can we actually appreciate some of the heterogeneity in HR status that might exist within these tumors? And so instead of pseudobulking to the patient level, we pseudobulked to the patient sample level. And once again, ordering samples by their true genomic HR status, we're able to see quite good concordance with the identifier predictions. However, when we looked at the actual proportion of cells coming from different sites, we were also able to appreciate that there were no biases or no sort of no sampling dependencies that we were kind of overlooking. But what was really interesting was that where there were multiple samples from different sites per patient, in cases where the identifier predictions were quite extreme, so a very HRP case or a very HRD case, we saw a low level of heterogeneity or variance in our identifier probabilities, in the probability of that sample being HRD. However, where the overall sort of probability was more intermediate, where it was close to our threshold at 0.5, we did see more heterogeneity based on different sampling types. And so this suggests that we're not just trying to binarize our cohort, but we may actually be able to appreciate some of the heterogeneity that exists within a disease. And so perhaps this tool might actually be useful, which was exciting. And so regardless of this, we found that there was an 85% of the site-specific HR statuses predicted by identifier were concordant with the patient level status. So it's okay if you only have bulk RNA sequencing data for this, hopefully. So in summary, I have presented Identify to you all. It's a gene expression model based on a differential expression signature that comes from known regions of copy number alteration. that means that we are picking up some of the genomic scarring that's used as a gold standard to predict this phenotype. I've tested this model in a held out cohort from the TCJ and found it to be accurate and to outperform other methods. I've tested this in an independent cohort, found it to be accurate, and I have now also tested its ability to be applied to pseudobulk single cell data, and we have many other results, but I have no time. And so these are my acknowledgements. If you're curious about this work, I'm more than happy to chat. That's a link to our preprint. It's been like conditionally accepted. I just need to write a rebuttal letter. I have had no time. And I'm very tired today, and so I don't know if it's going to happen today, but it hopefully will be out soon. And Identify has been submitted to Bioconductor, but nonetheless can be accessed at our group's GitHub. So if you're curious, give it a try. Be sure to roast it. I'd like to improve it. I'm sure there's room for improvement. So yeah, thank you.
Alex, a little bit, but just like one last round of questions.
Yes, Natalie.
So good with, that I love identifying. So you mentioned that HRD is not a once and then decided for the rest of the patient's trajectory, but it could change back and forth, especially through the addition of PARP inhibitors. Is there any data, publicly available data or augmentations to your model that you think would be useful into taking that into account?
You sound like reviewer 3. And I mean, to my knowledge, there isn't. We're trying to currently work with some of the groups at the Peter MacCallum Cancer Centre across the road from us to access some of this data. We feel that some of the cases where Identify suggests they are HRP, though their genomic signature or their genomic scarring score suggests they are HRD, we believe we can identify reversions in some of those cases. And so perhaps we're getting at something slightly more functional or more currently functional, but I'm not confident with the number of samples that we have. Like it could just be by chance, right? Like you can't base anything off of just a few samples. I would feel better if I had a properly curated cohort. So if you know of it, that data existing anywhere, I would love to do that work. But yeah, to my knowledge, yeah, at least not publicly available and known about. Hi.
Thank you so much for the talk. This is very, very exciting work. This is more of a wild thought. I think somewhere across the hallway or in a different floor for you at WHI, there's a group that has been working on the characterization of variant effects. So the MAVE team with Alan Rubin and others.
Yeah.
So they've been doing this multiple assays for variant effects, with a very heavy focus on cancer. And so I'm thinking that maybe your data set in the characterization that they have done can inform each other on further characterizing this particular type of cancer. I don't know.
Yes, I've had many chats with Alan about this. It's a little bit tricky with ovarian cancer because it's quite distinct from other cancer types in that it is largely driven by copy number change. We do see recurrent mutations, but the predominant signature is copy number change. And so we're looking at different ways that we can not only interrogate HR deficiency through transcriptomics and hopefully get a better understanding of functional tumor biology, but we're also now looking at other targetable features of the disease through the same method to try and get at functional subtypes in a little bit of a more meaningful way. But yeah, I've been harassing Alan and I will continue to do so.
I'm wondering if you've seen any correlation between neoadjuvant chemotherapy exposure and the results of your model, because I'm wondering if chemotherapy will affect genome scarring.
So I think it will. And so I don't know. So obviously, like, genome scarring isn't random, right? There are common fragile regions, but there are also regions that will be, sort of tissue type specific and unique. I suspect that it will. I've had I've got like 5 or 6 cases and the model still works. But that's 5 or 6 cases, it's nothing, right? Like that's not capturing the heterogeneity of the disease, and that's not enough to come to any sort of sound conclusion, within me at least. I can't do that. I expect that there are instances where this model will not be appropriate. So our cohort was largely treated in the traditional sense where they've had adjuvant chemotherapy following bulk or debulking surgery, primary debulking surgery. And so we haven't looked at that in enough detail, but it's something that I'm very open to. So yeah.
All right, so let's give a final round of applause.